Okay, this evening, Laurel Thomas is here to make a presentation to our group. She received her degree in environmental science in 2023, and that was the year that she was one of our chapter scholarship recipients. We're really pleased to have had her apply. And currently she is involved in seasonal ecological work while applying to PhD programs in applied conservation. Tonight she will present her research on our local toyon. So, Laurel.
All right. Thanks, Snowdy. Can everyone see my screen all right?
Yeah, I can see it.
Awesome. All right. Well, I just want to welcome everyone and thank everyone for taking some time this evening to listen to the work that I've been doing for the past few years. And it's all about toyon, which I know is a special plant to everyone. I think that's the name of your newsletter. This work was funded by your chapter, as well as just the state California Native Plant Society chapter. And so I'm just really happy to be back here and share the work that your chapter has helped fund. Before I get into myself and the work that I've been doing, I want to acknowledge the land that this research was done on, as well as the land that we're all joining from today down here in Los Angeles. Toyon, the plant, is a very significant resource to the Native American tribes of this region, including the Chumash, Tongva, Luiseño, Kumeyaay, Cahuilla, and Costanoan tribes. Toyon is thought to be the Spanish alteration of the Ohlone word toyon, and it's the only native California plant that's still called by its native name, which is very cool. And the research that I've done was conducted all throughout the California Floristic Province, which has historically over 500 distinct subtribes and today has 109 federally recognized tribes. So this work has a lot of importance to the indigenous people. Toyon is a very important and significant plant in California, and I just wanted to acknowledge that before getting started. And a little bit about me, who's talking to you today. I think it's important that you know where research is being conducted and who's doing it, because as much as we try to be as objective as we can, it's just not always possible to be completely objective. So my background and what has influenced the research that I've done in the past few years comes from me growing up in California and really having a deep love of California plants and ecosystems. I grew up in Northern California in San Jose. But I've been down in Southern California for quite a while, for about 4 years, give or take COVID in there. I studied environmental science at UCLA. And I did a lot of research in the Los Angeles area and the Santa Monica Mountains. I worked at the Friends of the Ballona Wetlands for a year. I did research up in the Santa Monica Mountains with the National Park there. And some of my toyon research took place in the Santa Monica Mountains. I had a common garden location there, which I'll talk about a little bit more. And now I'm back in the LA area and super happy to be here and talking to you all tonight, because the past year I've been doing a lot of seasonal work all over the US. You can see some of those pictures down there. So I'm happy to be back in LA and enjoying, this time of year here. And then the toyon project itself, which I'm super excited to share with you all tonight. I did this research primarily during my last 2 years of undergrad in my junior and senior year. And for the past year since graduating, I've been working on getting that written up and getting that published. And we are in the last steps of getting it submitted or of getting it published at the American Journal of Botany. And that should be out in the next edition. We're working on the copy editing with them right now. So if you're interested in the work that I've been talking about, you can keep your eyes open in the next edition of the American Journal of Botany. And the work that I was doing, like I mentioned before, was funded by the California Native Plant Society. So I'm just super grateful to be back here and get to share some of the work that you all contributed to. And just to make sure that these acknowledgements don't get lost at questions and at the end, a lot of people supported this project. I had a wonderful research advisor at UCLA who's now at the University of Connecticut, Rachel Prunier, and a lot of undergrads helped work on this project. My family was very supportive and came collecting and helping me with all of my plants. And the UC Natural Reserves were also a super great resource for this project as well. So now I'm going to jump into my project and talking about that. And I just want to remind you all that please ask questions, not like out loud right now, but write them down and put them in the chat. And I would be super excited to answer them and talk about them with you all. My goal for you tonight is that you find something new and exciting that you hadn't thought about before. And so feel free to, make sure to put those questions in the chat. So I want to talk about, I'll start about talking about why I did the research that I did, then talk about how I did the research, and then the results that we did find from the research. So the main reason why we did this research is that plants are very vulnerable to climate change, especially vulnerable because they can't move themselves very easily. So plants don't have very many options when it comes to bearing the changing climate. Like humans are much more easily able to migrate, adapt, put on sweaters, incorporate AC, but plants have to just stay in place, and they don't have as many options to contend with the climate change. And so the three accepted options that plants have are as follows in order to survive. So the main option that they have is migrating to a more suitable habitat. And this can happen naturally with plants naturally dispersing into areas that are more suitable through wind dispersal, and also the animals that pollinate them dispersing those seeds to different ranges that might be more suitable for them. And that can also happen with humans assisting this migration in restoration and other options by taking species and moving them to areas where we know that climate will be more suitable for them. The next option that plants have is they can change their phenotype, so how they look or how they're functioning to match the climate. And this is also known as something called plasticity. So this is happening within a plant's life, not across generations. And the plant might be growing deeper roots to reach the water table because it's drought times, or it might be growing taller branches to get more sunlight if it's cooler climate. So this is things that plants are doing within their lifetime, and this is one option that plants can do in order to survive and maintain their population. The last option that plants have is that they can adapt to the changing environment. And this is different than changing their phenotype, changing how they look within their lifetimes. This is over different generations. Certain adaptations that the plant has is being selected for and continuing on. And this will result in populations that are just better adapted to the changing climate. And this is happening over time, over generations of plants. And if none of these three options happen, then we do run the risk of plant populations being extirpated, so locally extinct. And this is just something to consider with the changing climate. And all of these are relatively understudied in plants. A lot of research in regards to climate change is really focused around animals, and just research in general is often focused on animals. And so there's not very many studies looking at how plants are responding to the changing climate and how we can take what we know about plants and their abilities to survive and inform conservation decisions. All of these different ways that a plant can survive are based off of 3 different causes of intraspecific variation. And what intraspecific variation is all the different ways that plants can be varied. So within a species, we can notice a lot of variation, and leaves might look different. They might be different heights. In California, especially, we have a lot of intraspecific variation. And there are 3 different reasons why there can be variability within a species. So think of it as how there are so many different ways humans can look. Plants, there are so many different ways within a species that they can be variable. And the 3 reasons that plants can have differences in their appearance is 1, because of genetic differences. And genetic differences, so our genes, our makeup, can be one reason why we're different from another. And this can be because of local adaptation, like I was talking about before, over generations. Plants are being selected to be a certain way to be more ideal for an environment, or this genetic differences can just be random genetic drift. Things are just happening randomly. Another reason why there can be variation is phenotypic plasticity. So I mentioned plasticity before, and those are things that are happening within the lifetime of a plant. There may be growing taller, growing thicker leaves, growing deeper roots. These are all things that are happening within a lifetime, responding to the environment, that are making them look different. And the last one are maternal effects. And this is equivalent to if the plant mom is more healthy or less healthy, and it's passing on their success or not unsuccess to its offspring. So a particularly healthy mom might make a healthier seed, and then their offspring would be much bigger and healthier, we might predict. And so these are the 3 different ways that we can account for the variation that's happening in plant species and humans. All goes for the same. These are things that are happening in all species. And these are very applicable to how our plants are surviving in climate change and how we're going to be making decisions for restoration. And it's very important to understand why plants are variable when making these decisions. So I'm going to bring it to restoration, and this is the main application of the work that I'm doing. And I think a lot of the conservation work that needs to be done for plants is in the restoration world. And it's important to think about the intraspecific variation, or how plants are varied. So historically, and even still, restoration managers assume that locally sourced plants are the best plants for a restoration project. And this is assuming that plants are locally adapted. It's assuming that plants for generations have been selected for ideal and optimal traits for that environment, and they're going to be doing better in that area. But this completely ignores a lot of other factors that could be going on for variation, and it also ignores climate change. So still assuming that plants are locally adapted, it doesn't necessarily account for climate change. So we need to consider climate change when we're thinking about restoration decisions. And this might result in sourcing seeds from regions that are going to match the predicted climate of a restoration region. Instead of just taking plants from the Santa Monica Mountains, propagating them and planting them in the Santa Monica Mountains, we might want to take plants from San Diego, because those are warmer sites, and bring them up into the Santa Monica Mountains, anticipating a warmer climate. And this would all rest on the assumption that those plants that we're taking for restoration are locally adapted, and they're well suited to the areas that we're taking the seed sources from. And both of these don't take into account that the fact that this variation might just be because the plants are plastic. It might just be because the plants are in San Diego or in LA that they look different and are acting different, and their genome, their genetics might not be that different from each other. And in this case, when plants are plastic and it's based off of their environment that they're very variable, it might be better to source seeds and source plants for restoration from a wide range of areas. If plants are going to do similarly, no matter what environment you're taking them from, because they're plastic, they'll just respond to the environment no matter what. it's better to source from a wide range of regions, because then plants will have more genetic diversity and increase the chances of success that one plant's going to survive climate change and do a better job. So all of these things are things we were thinking about when thinking about toyon. Looking at what is the reason for the variation that we're seeing in toyon and thinking about it in terms of restoration. So that brings us to why we have all these questions about restoration, intraspecific variation, how plants are going to survive climate change. And we wanted to ask these questions with a plant that was significant and important. And so we started in California. California is a great place to do studies like this because it's a biodiversity hotspot, as we all know, and it has really rich plant endemism and just a really high amount of plant species and a wide variety of ecosystems. So this is allowing our research to be applied to other biodiversity hotspots and other various ecosystems because we have so many ecosystem types. At the same time, California is experiencing a lot of habitat loss and fragmentation and is experiencing large results of climate change. So this is making our work a lot more important and critical because we have this juxtaposition of really rich biodiversity and also really high rates of urbanization and climate change. And there's a lot of restoration work and conservation work being done in California. So that makes studies on California species really applicable for all of the restoration managers and nonprofits that are doing restoration and conservation work here in California. And we picked our study species, toyon, because for many reasons. The first one is that toyon is very ecologically important. It's found in many different areas of California, in oak woodlands to coastal chaparral. I've even seen it tucked under some redwoods one time, which is really it's not supposed to be there, but it can do a lot of crazy things. And it's ecologically significant, being one of the few things that's fruiting in the winter, and it's a very important food source for birds, and it's also an important food source for Indigenous people. Another reason we selected toyon is because it is a very crucial species for restoration. Toyon is fast growing, very hardy, and makes up so many important parts of different ecosystems. So this is a very important study species to consider, and it will have a huge impact on all the restoration going on in California. And the last and final reason that we are asking these questions about toyon, about why it's very varied and how this applies to conservation decisions, is that toyon is highly variable. It's doing a lot of weird things across California, and it's also very widespread. So you can see here in these pictures that in the first top picture on the left, toyon's leaves are almost entirely smooth here, which is very interesting. And in the bottom, the teeth margins, the teeth along here, is very spiky. So that's a high variation across this one species. And then this picture gets me every time. You can see here there's green berries, almost red berries, and then at the bottom there are flowers of that toyon. And so this is happening all on one plant at one time. So you can see that it has a lot of variation in reproductive timing, and that just one plant itself has this many things going on. It suggests that there's a lot of plasticity going on because it can respond to its environment in weird ways. And toyon is not the only plant that has this wide of a range in California, but it is rather unique compared to the whole world. That toyon makes this range all the way from almost Oregon down into Baja California. It's found on the Channel Islands and Catalina, and it makes its way up even into the foothills of the Sierra Nevada. So it has a very wide range, and it allows us to ask a lot of interesting questions and have a really good study sample across the state to ask questions that we want to know. And so these are the main two questions boiled down from everything that I was talking about in terms of climate change, restoration, and intraspecific variation. So what we really wanted to know is toyon widespread and highly variable in California because the populations are adapted to the local environment, or is it because the plant is phenotypically plastic, or is it a combination of these two? So basically asking the question, is it nurture, how the plant's environment is, or is it nature, how its genetics are? And then taking these findings, we wanted to think about the implications for conservation management and restoration. So where should these seeds be sourced for restoration projects for toyon? So this is the how we did it, how we answered these questions. And so these are the methods of our toyon project. So it started with collection of herbarium samples and leaves. And so these were collected all across the state of California. You can see here there are 286 locations where leaves were collected. And this was done in collaboration with another lab at UCLA, and a lot of travel across California was done. And with those leaves, all of them did have DNA extraction done in order to look at genomes. And that's not a part of my specific project, but it can be potential expansion. And after this initial collection of leaves, we then collected from 70 of different individuals. And in the end, after all the propagation and germination happened, we had 37 maternal lines to do a study on. So this is the propagation process of the seeds. We extracted over 4,000 seeds from those individuals across California, and we germinated those seeds in Petri dishes. We started with half of them in the fridge and half of them in like a cool, dark box, and very quickly ended up moving them from the cool, dark box into the fridge because of mold. Even though we sterilized and used bleach on all the seeds, we still were contending with mold. So the main strategy for germinating the toyon seeds were in the fridge. And we then outplanted them in the greenhouse at UCLA in randomized blocks, enough to have 12 individuals per line. So we had 12 blocks and randomized all of the individuals within them to make sure where they were in the greenhouse wasn't affecting how they were growing. You can see here some cute things of our toyon propagation. In this first picture on the top here with the seed, you can see kind of this gel that was around the toyon seeds, which was really interesting, kind of like a chia seed. They got really like puffy and jelly-y. You can see it like kind of sticking to the to the petri dish, which was really cool. We were thinking that this was probably an adaptation to drought and variable weather, because California has those wet winters and dry summers. So we were thinking that, you know, this was probably an adaptation to retain moisture. But that was pretty cool. And you can see here some really cute little toyon cotyledons. They start off so small for how big they get, and some cute first leaves here with the toothy teeth. My research advisor would go down to the greenhouse when she's having a bad day and look at our toyon babies. So they're pretty darn cute. And yeah, here's just more of the cute plant babies. We had a little army of about 500 toyon in the greenhouse, and this amount of growth took about 2 to 3 months, so they grow pretty fast for woody species. I thought you guys would think these were cute, all of the plants. Yeah, so after all of this propagation, this was setting the stage for establishing common gardens. And so common gardens are really important for looking at intraspecific variation in plant species because it allows them to be grown in a common environment, and we can really filter out whether these plants are doing these things because they're plastic, are they responding to their environment, or are they going to be different no matter what environment you're growing them in because they have genetic differentiation. So putting them into common gardens allows us to really filter out, are the differences that we're seeing across species because of their environment or because of their genes. And for this reason, this is why you need 2 common gardens, so you can compare one environment to another environment and see if there are any significant effects of that garden. So for our experiment, we had 2 common gardens, one in San Jose, California at the Blue Oak Ranch Reserve, which is a UC reserve, and another one at Scent Ranch Reserve, which is actually in the Santa Monica Mountains. And at each of these gardens, we put 6 replicates of each of the 37 maternal lines. We had a little over 200 plants at each of these gardens. And we were able to take the data of each of these gardens, looking at their traits, and to see if there were any plastic effects of our traits, or to see if there was genetic differentiation. This is all of the plants that were planted at the Stunt Ranch in the center here. We put them in my research advisor's Prius, and they're looking pretty cute in there. And then they all got planted out in the garden down here at the bottom. And just a little aside, the UC Natural Reserve System is a really incredible resource, not just for people affiliated with the University of California, but for anyone who's interested in education, research, or just being outside. A lot of research is conducted at all of these 41 reserves, and there's one right here in Santa Monica up in Calabasas along Stunt Ranch Road. If you haven't been there, I really recommend checking it out. There's a hiking trail that goes through it, so you can hike through part of it, which not all of it's open to the public, but part of it is. But often UC reserves host educational events, and they're more than happy to have events for different organizations and have research and study happen there. So they're just a really fantastic resource, and all of the reserve managers are just really wonderful. So I really recommend if you're interested in having like a retreat or an educational visit, or do a research study of your own, these are really fabulous resources. And yeah, just highlighting again where the 2 UC reserves were that I had my common gardens at, but a lot of these other reserves I collected toyon berries and leaf specimens across the state. So they're just really great resources. So back to the common gardens themselves. They were constructed in 2022 during my junior year of college, and taken care of primarily over that summer in between junior and senior year. We did a lot of work to prep for these common garden sites. We did some rototilling down in Stunt Ranch because the soil was super clayey. And after we prepped the trenches for the plants, we covered with cardboard at both sites to keep the moisture in and the weeds down. We also constructed chicken wire fences with T-posts around it to keep out herbivores. And you can see here milk cartons over the little toyon plants. I learned this method actually from the Santa Monica Mountains restoration project. They like to put them over to protect from herbivory, and it also works as shade from the plants. We were planting these in the summer, which is not an ideal time to plant in California. So we had to think about how we were going to keep these plants from dying all right away. And these milk cartons were a really great resource. You can also see here, they were so nice and cool underneath that. We had a little family of toads living underneath one of them. There's just one of the toads there. And then I was letting them live there, but then they uprooted one of the plants. So then I kicked them out, and there's a sad toad down there at the bottom on the hose. So all just fun adventures of having a large field experiment, and yeah, dealing with toads. That was a fun adventure. Yeah, and so all of the data that we were collecting in order to answer our questions about variation and how these plants are going to respond to climate change and what this means for restoration were primarily leaf functional traits. Leaf functional traits are really important tools for research because leaves are really important to a plant. They facilitate a lot of interactions between the environment and the organism, and they're very easily altered. A plant can grow more or less of them and change the thickness and the area of them, typically they're usually pretty plastic and malleable parts of the plant. And leaves are also very easy to measure. You can take a leaf from a plant and the plant will be fine, most likely. They're just very useful traits to measure. And so the traits that we were focusing on were the stomata, the specific leaf area, which is how the area of the leaf compares to the thickness of the leaf. And then we were really, really interested in the toothiness um, of the toyon leaves. As you saw in one of my earlier slides, there was a lot of variability in the leaves that we were seeing in the wild. So really smooth margins to really toothy margins. And we were really curious to know if that might be a local adaptation of toyon, or if it's just a plastic trait, or it's just randomly happening. And then for the data analysis, just a quick little chat about that. All of that was done in RStudio, which is a really common platform that we use for stats in ecological research. The primary tool that we used are linear models. So we're just looking at how one thing that we're interested in. So my example here is, how does leaf area change based off of these other variables? And it's really easy. You just plug it into the code. So we're wondering, how does leaf area change based off of temperature, based off of other climate variables, and the maternal traits that we did have, because we had measurements for the leaves of the moms and measurement for leaves of the offspring. And so that's just a little bit of how we looked at it in RStudio. And you're welcome to ask me more about that in like an email or whatever afterwards. And then we used to get the climate data, this awesome data set WorldClim. So for pretty much any point in the world, you can extract 19 different climate variables on precipitation and temperature and seasonality. And then you can have that for your collected location. So it's just a really useful tool to be able to look at climate trends. And all you need is the coordinate to access that data. So some results now. We wanted to know, so the first part of our data analysis and collection was just primarily on our maternal line. So those are 286 individuals that we collected from. These could just answer questions. Is the variation we're seeing a pattern at all? Is the variation we're seeing a result of climate, or is it just random variation? Are there no patterns at all? And we wanted to establish this first to see if there were results of the environment on the phenotype of all of these maternal lines, because otherwise our questions aren't very useful. If things are just random and variable, then everything is just probably genetic drift. Everything's random. There's no local adaptation. There's no plasticity. So we wanted to establish this first. And we did this by looking at those traits that I was talking about for leaves and also seed mass, and seeing if those were related to different environmental variables like temperature, precipitation, and elevation. And we did find that trait variation is correlated with the environment. So we were finding relationships between different traits and the environment they were sourced from. Some of the things we found is that seed mass decreased in areas with more precipitation. We also found that specific leaf area increased in areas with more precipitation. And we found a lot of trends primarily with teeth number and teeth length. So how many teeth are on the toyon and how long are those teeth? Here, all of these graphs are all about tooth length and teeth number, and how they're relating to different temperature and elevation traits. So this is suggesting to us that a lot is going on with teeth number and length, and it could either be local adaptation that we're seeing, or it could be plasticity. And we can't answer why the variation is there until we look at the data from the common gardens. But we can say that there is interesting intraspecific variation that's happening, and it's correlated with the environment. So it is worthwhile to do a common garden study to investigate these patterns even more. So here you can see tooth length and teeth number have significant relationships with the environment. And so that's very interesting, and it's a good sign to continue moving forward with the common garden study and to look at anything we're seeing there. And so we have to take this question and move it into the common garden to understand, are these trait differences heritable and adaptive in the common garden, and to what extent? And so here are the common gardens again. And we also collected data in the UCLA greenhouse and in both of the common gardens. One interesting finding that we had, we were able to look at heritability. So how much of a mom's genes are passed on to an offspring's. And we were able to do this because we had data on the maternal leaves and the offspring leaves. And what we were able to find is that the average length of teeth on the leaves is a heritable trait. And it's pretty strongly heritable, actually. This is a very high amount of heritability with a slope of 0.4 and 0.3. Those are really high levels of heritability. So it's very interesting that tooth length is being passed on from mom to offspring. And we were able to collect that from our common gardens and our maternal line data. We also found that teeth are correlated with their source environment. So in the common gardens, we were finding a lot of patterns with teeth number and the length of the teeth being predicted by where they were sourced from. So we're seeing an important relationship between how the teeth are presenting in the gardens to where they were sourced from. All of that, so teeth are heritable, and teeth have a significant relationship with where they were sourced from. And we also found that teeth number and length have a significant effect on the fitness of these traits. So plants are doing a little bit better in most cases when they have longer teeth and more teeth. So we're seeing that they're growing a little bit faster, they're growing a little bit taller, and they're growing some more branches and leaves. So you can see here in this table where there are stars is like significant results in our stats. And so more teeth and longer teeth are in some of the different environments that we were collecting this data are resulting in more fitness. We also found in terms of plasticity, so this is kind of a confusing figure, but these are estimations of plasticity, and the lower, the closer the bar is to the dotted line in the middle, the less plastic it is. So you can see here, tooth area and tooth length are pretty close to this dotted line, and we can say that it is less plastic than these other traits. They're a bit lower on average than these other traits. So all of these things that we found together provides pretty strong evidence that toothiness, or how toothy a toyon leaf is, an adaptation to local conditions. So we found that teeth length is heritable and less plastic than leaf and other traits. We also found that teeth length and number are correlated with their source environment, and we found that toothiness increases with height, relative growth rate, and total new leaves. So these are all strong factors to supporting that variation in the variation that we're seeing in toothiness across California is because of locally adapting to those conditions, not just plasticity responding to that environment. On the flip side, from our toothiness, other traits are quite plastic. So we see here the average major tooth length and tooth area is low in plasticity, but these other ones are pretty far from the center line. And so this is leaf area, leaf number, relative growth rate. These are pretty plastic traits. And this is not too surprising because things like leaf area and how fast a plant is growing, that makes more sense to be a direct response of the environment and not something that our genes are telling us about. And this is just also showing really the big difference in traits between this right here is one common garden, and this is average. On each side is an average of that trait value, and then it's a line drawn to the other average of trait value in the other common garden. So you can see here that there's quite a big difference in each trait from maternal line from garden to garden. So we're really seeing high amounts of plasticity in a lot of these traits. The average of trait in one garden is a lot different than the average of trait in the other garden. And that's how we measure and look at plasticity. So that's just a little graph showing how that's working for all the different traits. So even though we found really strong local adaptation for the toothiness, it still didn't strongly affect fitness by the source location. So we weren't seeing an effect of where the plant was sourced to how it was doing in the common gardens. So we conducted survival analyses and models looking at the fitness components in R, and we weren't finding any significant results of where these plants were sourced from and how they were doing in the garden. And so this is just important to note that plants were not more fit when they were sourced from environments more similar to the common gardens, which is what we would expect if these plants were really heavily locally adapted. And we also found that plants did not survive more when they had more teeth, even though they were doing a little bit better with relative growth rate and leaf number and branching. So this is just showing that even though we did find this really cool local adaptation in the toothiness, this isn't a big enough effect on the fitness of the plant and the success of a plant in establishing. And so overall, it doesn't really matter where we're sourcing these toyon individuals from, and they still have similar rates of success in the gardens. So these are just the big conclusions to our overarching question. We were wondering the intraspecific variation of toyon. So is toyon widespread and variable because populations are adapted to the local environment, or because the plant is plastic, or is it a combination of those two? So our main takeaways from this was that toyon traits are highly variable and they vary with the environment in predictable ways. And we answered that with just looking at the maternal lines. We also found that toyon is plastic for most of our leaf traits, and toyon is locally adapted. We saw this in its teeth, but the source environment does not play a big role in its fitness. And then in terms of this implication for conservation management and restoration, even though we found this local adaptation in the teeth, I think we can make the claim that toyon can be sourced from any location for restoration, because we did not see any significant effect of where the toyon was sourced and its survival in the common gardens. And this is a big benefit to restoration of toyon because it will provide more genetic diversity, and it will just make restoration easier in terms of sourcing seeds for using toyon as a restoration, or where you're purchasing your toyon plants from. You don't have to be as concerned for where it's being sourced. And another takeaway is just toyon is a very hardy plant. It can do a lot of crazy things. It can grow in a lot of weird places. I'd be almost into the desert and see a toyon. I'd be on the edge of a cliff over the ocean and there'd be a toyon just doing its thing. It grows in so many different environments, and it did remarkably well for us planting them in the middle of summer in California, which is not the ideal planting time. And they even survived, I couldn't believe this, but in February of 2023, there was a snowstorm, and this is on Mount Hamilton, the tallest peak in San Jose or in the Bay Area. And you can see quite a few inches of snow on one of my common gardens, which is pretty funny to see that picture. And when I came back to visit them, they were doing fine. They didn't seem to care that they got snowed on, which I thought was really surprising. They're just a pretty tough plant. Yeah. and going for tough plants, these are some of the remnants from my project that lived in the greenhouse in my backyard in San Jose. They're doing just fine. So this is about 2 years later. They're still growing. And I just want to quickly talk about some expansions to this work that could be done, not by me. I'm about to wrap this project up, but there's a really cool project that's being done. If you haven't heard about it, it's the California Conservation Genomics Project. And this is a collaboration between UCs, CSUs, and other institutions all across the state of California. And it's looking at the genomics and the landscape genomics of a whole bunch of species across California. So not just plants, but invertebrates, birds, trees, lichens are being collected across all of California, and there are reference genomes being created, and then they're looking to see if there are any locally adapted genes in these plants. So my work with toyon started in this area. It started with me extracting DNA and looking at the genome of toyon, and this could be paired with my common garden data to really look and see what the genes are for the things that we found that were locally adapted. So that would be toothiness of the toyon. There might be some genes in there that we could find with this project. And then just like overall, making generalizations of this kind of work. Common garden experiments are very time intensive, and it's not feasible to do this work for every single plant that we're interested in. And I think that we should know more about the plasticity and variation of plants in order to make better restoration decisions. So I just want to think about ways that we can generalize these studies and make conclusions across species and across species types in order to prevent common garden, in order to not have to do common gardens for every single plant species, because we don't have time to do that with climate change. And I went to the seed sourcing talk at the California Botanic Garden that was recommended last time, and they were talking about things similar to this, about whether we should be locally sourcing seeds or making mixtures of seeds if they're plastic. And a similar conclusion was made for purple needlegrass as we made for toyon, that it can be sourced from a broader range and not specifically locally source those seeds. And I'm just wondering if we can make these generalizations for a lot more species to just save time and energy in research. And that brings me to the end of my talk. I really appreciate you all having me here today, and I hope you learned something cool about toyon. And yeah, thank you. I see someone with a question already.
Yeah, you ready to take questions?
Yes, I'm ready.
Okay,
So,
cool. So I have a couple.
Laurel, do you want
uh,
to unshare
Oh
your,
yeah, I can stop
so
sharing.
we can see people? Yeah.
Yeah, good idea.
So thank you very much for that presentation. That was very informative and I really appreciate it. Two quick questions for you. Number one, I've always understood that the teeth on the plant is generally a response to, predation, grazing kind of a thing, that's why it evolved. And, the example often is like with Prunus salicifolia, you have the hollyleaf cherry in the mainland that has teeth, and you have the Catalina cherry on the island that doesn't, because until recently there were no, grazing species out there. So my question is it outside the scope of your study, or was there any correlation whatsoever to areas that have concentrated grazing species and the existence of those teeth, and if that was a factor in survival? And the second question I have, which I'll throw out so I can be quiet, is my my assumption is from your work on the plasticity of these is that this is actually an argument against adapted human intervention and adapted climate migration, because the species throughout its entire range seems to have equal robust nature from the seeds, no matter where you're planting it. Does that am I drawing the right conclusion from what you're saying?
Yeah, and I guess like to answer your second question, I wouldn't make that generalization for other species, but for toyon, I wouldn't recommend sourcing plants from San Diego and bringing them up to Santa Monica Mountains, or
Got it. Thank you.
that would be a waste of resources and time to look at predicted climates and assisted migration. So for toyon, yes, yeah, I wouldn't recommend that, and would just recommend sourcing as widely as possible, and maybe just thinking about shifting the whole population up north, more north, but not sourcing from certain areas, if that makes sense. And then in terms to leaf margins, there's like a huge can of worms that you could go down, which I did go down doing research for writing my paper. So toothiness is a defense against herbivory, but there's also a lot of other reasons why plants have teeth, and there's a lot of unknowns about it. Something that's really interesting is the amount of teeth a plant has is actually like a global pattern based off of temperature, so and latitude. So there's like this, it's since like 1916 we've had this pattern, like the closer to the equator you get, the less margins a leaf has, the more smooth they get. So think of tropical plants, they have really smooth leaf margins, and then as you move away, it starts to become a lot more margin. So in our temperate areas, we have a lot more lobed and toothy leaves. So that's something that's really interesting, and something that we were thinking about in terms of just that relationship between temperature and teeth margins. Paleobotanists actually use like how toothy a leaf is to see like what the climate was from that fossil. So it's just really interesting. I never knew how important leaf margins were until I started researching toyon. And so no, we didn't look at herbivory in seeing if that was causing local adaptation. We really only looked at like elevation and temperature and precipitation and those relationships with teeth margins. Yeah, we didn't think about herbivory at all. We were just mostly focused on preventing herbivory in our gardens, so that the plants that get eaten. But that's
Thank you very
interesting to
much.
think about.
Yeah.
Well, there's some questions that have come in the chat. I'm going to take them backwards. What are some general site differences, example, climate, soils, elevation between blue oak and stud ranches?
Yeah, thanks for asking that question. So Blue Oak Ranch is at a higher elevation than Stunt Ranch, and it's a bit cooler and receives a bit more precipitation than Stunt Ranch. So they were selected for two reasons. They're like on the extreme edges of toyon's range for the most part, so that could really help us answer questions. And then also for ease of access, one was near UCLA and one was near San Jose. Yeah.
How did you clean the seeds before taking their mass?
Yeah, that was a project. So we purchased like sieves that we were planning to like push the seeds through. And then as I started doing that, I noticed that I was damaging the seed coat. So began manually extracting them. So I watched a lot of TV and broke open the toyon seeds and pulled out the seeds. So yeah, we did not find like a efficient way to extract like thousands of seeds, just because the seeds are so small and they were really delicate. When pushing through a sieve, they were getting scratched on the seed coat a lot.
Are there other aspects beyond survival rates that we should consider when deciding where to source restoration from? For example, palm production, food for animals, insects, etc., plant size, crowding out other local plants.
That's a really interesting question. Like I guess we were thinking about these questions purely in terms of survival and fitness. Like, is a plant adapted to the environment it's being sourced from? But those are really interesting questions to think about. Yeah, I feel like you could think about a million things then and start looking at all of that before you start restoring. I feel like our primary concern was climate change, will it survive? Is it fit there? But that's really interesting to think about. I'm planning to go to grad school for restoration ecology, so maybe I'll think about some of those questions. That's cool. Thanks, Michael.
What happened to all those baby plants? Where are they?
So some of them that were in the greenhouse, those continued on. Some of them are planted at my house, at my aunt's house, but the ones that were in the common garden, my research advisor was quite insistent that we biomass them for data collection. So we also could not leave the toyon in their common gardens because since they were from a whole bunch of different environments, the reserves didn't want to have to worry about them reaching reproductive age and mixing with other toyon on the reserves. So yeah, the plants in the common gardens were uprooted and biomass. So I put them into a drying oven, and it was very sad. But I guess that's how research goes sometimes. Yeah.
Here's another question. Have you tried to use climate models to see where the current habitat conditions exist in 2050, 2070? P.S. I'm not denying the plasticity of toyon. There is an established one at Springs Preserve in Vegas and at least 2 at Desert Museum in Tucson. But those are in cultivation. The natural
That's cool.
system is different.
Yeah, that's cool that they're surviving in Arizona and Vegas. That's what I'm saying. They're a pretty tough plant. Yeah, I have looked at like the current habitat conditions in like 2050 and 2070, and that was something like we were considering when like before we had gone through the data and wondering if we were finding this local adaptation with the leaf margins, but we hadn't really figured out if where they were sourced from had effect on fitness. So we were looking at those to maybe think about where to source seeds from. But we didn't end up going down that route further when we were like, oh, toyon is just so plastic. But in the future, I definitely use that for deciding where to seed source. Yeah, it's a great resource.
Well, I had one of my own questions because I think I covered the ones that were in the chat. And since I've been dealing with native plants for many years, there's always been this, like, it's the Bible kind of statement that we should also always in restorations source our plants from the same watershed. But I'm having a feeling what you're saying is because of climate change, this may be not the accepted practice in the future.
Yeah, no, it's definitely like in all the restoration nonprofits I've worked with, it's always just like we collect from the watershed. I've seen that before in the nurseries, things are organized by watershed. And when I've done restoration projects myself, I always made sure I was sourcing from Theodore Payne or somewhere local to make sure I was, yeah, doing the same thing that you're saying. No, I think it's becoming a lot more disagreed with that is the best option. And I feel like that's for 2 reasons. One, because of climate change, that locally sourced plants might not be the best suited to survive in the future. And then also that completely ignores plasticity and plants like, especially grasses that are wind dispersed and aren't really locally adapting to an area as much. It's ignoring that plants could be just really plastic and doing whatever their environment is telling them to. And we might be resulting in like genetic bottlenecks and a lot of inbreeding if we locally source things. So it could be much more beneficial to, yeah, source from wider than just the watershed. But yeah, I know it's kind of like, it's very different than what's been, yeah, restoration Bible for a while. Yeah.
Okay. I really didn't understand. You had 222 plants, correct? Some give or take. Did you kind of distribute those between the 2 sites at random?
Oh yeah. So in each common garden, there is 222 plants,
Oh,
and
in each garden.
yeah,
Okay.
and it was completely randomized. We had all the maternal lines, and then we sent 6 plants from each maternal line into the 2 gardens, and that was completely random which plant got sent where.
Okay. So you really didn't then have like a comparison between the 2 sites, right? If it
The
if this site
comparison,
had the same plants as the other site, then I don't know. Okay.
the comparison was maternal. We had 6 from each, so there's 12 in the maternal line, and we sent half of them to one garden and half of them to the other. So
Okay.
the
All right.
same, each mom sent half of her kids to one garden, half to the other. So yeah, we were able to compare. Yeah, there's a lot of plants getting moved around. So yeah,
Okay.
little crazy.
And I had a question. I got a little confused about the concept of seed mass.
Yeah, seed mass, we weighed how much each seed was. Yeah.
Did you have any seedlings set flowers and fruits? If so, how many?
No. So yeah, there's the picture of the toyon in my backyard. I'm trying to do the math. I started growing them all so they're about 2.5 years old, the ones in my backyard. Still no flowers or fruits, but
Mm.
I don't know if I haven't grown toyon from seed before, so I don't know if that's long or short, but I think it should be making flowers or fruit soon. But like I mentioned before, and why all of the toyons got destroyed in the common gardens is that the reserve managers didn't want flowers or fruiting to happen, to have mixing between all of the different new populations and the populations there, just in case we found it was super locally adapted. Yeah.
Okay. Were there any other questions? Did I miss any? I thought that I caught them. Okay. Well, Laurel, I really thank you so much for all the work that you've done. And we look forward to your publication. I hope you'll let us know when that actually gets published. And good luck with your PhD work. I'm sure that will happen for you. So,
Yeah, thank you, everyone. Thanks for all the awesome questions.
all
Thank you
right.
so much. Great.
Yes, we really appreciate that you applied and that we were able to give you a scholarship too. It's really,
Yeah.
it's a good feeling from our chapter that we're able to do that.
Yeah, it was very meaningful. I very much appreciate it. Thank
Okay,
you.
thank you.
Mhm.
All right, guys. Thanks for attending, and we'll see you in the future.
Thank you.
Bye-bye,
Bye.
guys.
Thank you.
All right, ending the program.